involvement of any metabolic energy. Cations have also been shown to stimulate salt respiration. Selective uptake of ions cannot be explained by this theory. Now, after cytochrome pump theory, the mechanism involving ATP given by the Bennett and Clark, that is the protein lecithin theory. The theory advocates that a phosphate group in the phosphatide act as an active center for binding the cations. So cations binds at the phosphatide center of lecithin, whereas the anions bind at the choline group. The complete lecithin molecule, when combined with anion and cation, moves inside and then releases phosphatidic acid and choline due to the action of the enzyme lecithinase. Cations and anions are now released into the inner space. Now the lecithin is regenerated from the phosphatidic acid and choline by the enzyme cholinesterase and cholinesterase involving the participation of ATP. So this is how the involvement of ATP being explained by Bennett and Clark in his theory, that's known as protein lecithin theory. Now, there are certain protein carriers that act as channels for the transport of ions. Protein embedded in the lipid bilayer act as carrier, and then it forms complexes with the ions on the outer surface. Then rotation occurs at 180 degrees, and this rotation results in the release of the solute to the inner side. This is how protein carriers helps in the channeling process of the mineral salts from outer surface to the inner side of the membrane. This theory, the carrier concept involving the rotation of protein solute complex, has been criticized by Singer. He advocated that an amphipathic molecule like protein with its polar region to the surface of the membrane and non-polar region embedded in the lipophilic region will not rotate in the membrane. Now next theory, that is given by the Singer, bridging proteins. The bridging proteins acting as channels through membrane. The model proposes that the integral proteins that occur in the form of bridges in the entire membrane act as channel through which the substance is transported. The extrinsic proteins bind specific ions or molecules. The peripheral protein then attaches to the integral protein, that containing a hydrophilic pore. Finally, a conformational changes in the protein takes place that causes the release of the solute across the membrane. That is depicted in the picture, how the extrinsic and intrinsic proteins and their conformational changes resulting in the movement of mineral salts from outside to the inner side of the membrane. Now come the various factors that affect salt absorption. certain factors are external and certain are internal. External factors being temperature, pH, light, oxygen, and interaction. And the internal factors